Good morning, students. We will continue the topic protein metabolism, subtopic metabolic pathways of glycine, where we are seeing the synthesis and catabolism of glycine. Along with this, we are also studying the role of glycine in biosynthesis of various specialized biomolecules and disease associated with glycine metabolism. So first is synthesis of glycine. Glycine is a non-essential amino acid. So first glycine is synthesized from serine. If you see, I have shown the structure of serine above. When CH2OH group of this serine is removed, it is converted into glycine. So enzyme required for this step is serine hydroxymethyl transferase. Now in this case the group CH2OH is removed. This CH2 group is accepted by a coenzyme called as tetrahydrofolic acid, THF. This tetrahydrofolic acid is made up of three compounds: pteridine, PABA and glutamic acid. I have shown the structure of pteridine below. Now this CH2 group binds with the two nitrogen atoms, 5th and 10th nitrogen atoms of tetrahydrofolic acid. Therefore, this tetrahydrofolic acid is converted into N5N10 methylene tetrahydrofolic acid. So in this way, the serine is converted into glycine. So in the exam, you can write down this reaction directly. No necessity to draw the structure. This is about tetrahydrofolic acid. So this tetrahydrofolic acid is a coenzyme synthesized from folic acid, which is a vitamin present in our diet. The figure is shown on the top. Now this folic acid undergoes reduction with the help of enzyme folate reductase, and for reduction 2 hydrogen atoms are donated by NADPH, and these 2 hydrogen atoms are added between 7th and 8th atom of pteridine ring of tetrahydrofolic acid. So it is called as 7,8 dihydrofolic acid, which undergoes further reduction by same enzyme. Again, the donor of hydrogen is same, NADPH. Now the 2 hydrogen atoms are added between 5th and 6th atom of this pteridine ring, and the dihydrotetrahydrofolic acid is converted to tetrahydrofolic acid. So this is the synthesis of glycine from threonine, where the enzyme threonine aldolase acts on threonine amino acid, which has 4 carbon, and break it into two compounds. First is glycine and second is acetaldehyde. Each have 2 carbon atoms. Third way of synthesis of glycine is with the help of enzyme glycine synthase. This glycine synthase enzyme combines carbon dioxide, ammonium ion plus methylene tetrahydrofolic acid, or N5N10 methylene tetrahydrofolic acid, and synthesize a glycine. Here methylene group from tetrahydrofolic acid is added to the glycine and tetrahydrofolic acid is free. This reaction is a reversible reaction. During catabolism, the glycine is catabolized into these 3 compounds from which it is synthesized. Next method is synthesis of glycine from glyoxylic acid. Here this reaction is also a reversible reaction. In this reaction, glycine is converted into glyoxylic acid during catabolism, but during synthesis this glyoxylic acid is converted into glycine. The enzyme is alanine glyoxylate aminotransferase. Basically, glyoxylic acid is a keto acid. Here it combines with glutamic acid. This glutamic acid donates amino group to this glyoxylic acid, and after addition of amino group to this glyoxylic acid, it is converted into glycine. This is an example of transamination reaction. So pyridoxal phosphate is the coenzyme in this reaction. Now we will see catabolism of glycine. The previous reaction that we have studied, if we reverse that reaction, then glycine is converted into glyoxylic acid. Here glycine donate its amino group to alpha ketoglutarate to synthesize glutamate, and after removal of amino group, glycine is converted into glyoxylic acid. The enzyme is same, alanine glyoxylate aminotransferase. So this is the first method of catabolism of glycine, that is by transamination. So by this method, glycine is synthesized as well as catabolized according to the need of the cell. Second method of catabolism of glycine is by oxidative deamination by glycine oxidase enzyme. So this enzyme acts on glycine, removes amino group along with removal of two hydrogen atom. These two hydrogen atoms are accepted by a compound called FAD, which then reduced to FADH2, and here the amino group is removed in the form of ammonia. Here there is no acceptor of that amino group is present in this reaction. So this reaction generates free ammonia plus glyoxylic acid. Now we will see in short about glyoxylic acid. Basically this glyoxylic acid is synthesized in our body from catabolism of two amino acid. First is glycine that we have studied by two pathways, that is transamination pathway, where enzyme is alanine glyoxylate aminotransaminase, and second is glycine oxidase pathway, and third is from a serine with the help of enzyme serine dehydratase. Now this glyoxylic acid is later on converted into oxalate. Basically before converted into oxalate, this glyoxylic acid is also converted into fumarate and malate. Malate enters into the TCA cycle. Fumarate is converted into N10 formyl tetrahydrofolate and carbon dioxide, and oxalate is excreted in the urine. So this is about the glyoxylic acid which is formed from the breakdown of glycine. Next, catabolism of glycine with glycine synthase enzyme that I have told you. This enzyme catalyzes a reversible reaction in which it can synthesize glycine as well as it can break down the glycine. When the glycine is broken down, the carbon dioxide, ammonium ion and methylene tetrahydrofolic acid is formed. The enzyme glycine synthase is also called as glycine cleavage system, and coenzyme is tetrahydrofolic acid, which act as a acceptor of CH2 group present in the glycine. So this is the major pathway for breakdown of glycine. Now we have studied glycine synthesis as well as catabolism of glycine. Now we will see metabolic disorders of glycine. Three disorders of glycine metabolism are seen. First is hyperoxaluria, glycinuria and hyperglycinemia. We will study one by one. First, hyperoxaluria. It is of two types: primary hyperoxaluria and secondary hyperoxaluria. Primary hyperoxaluria, the enzyme defect is alanine glyoxylate aminotransferase. Normally this enzyme is located in peroxisomes of the cell, but in this disorder of primary hyperoxaluria, this enzyme is located in cytoplasma and hence it is inactive. As this enzyme alanine glyoxylate aminotransferase is inactive, so it will not form glyoxylic acid by this pathway, but at the same time it will not synthesize glycine by reversible reaction from glyoxylic acid. So the basic defect in hyperoxaluria or primary hyperoxaluria is glyoxylic acid is not converted into glycine. As I have told you that glyoxylic acid is not formed only from one pathway, it is formed from 3 different pathways. So if the first pathway is blocked, glyoxylic acid is synthesized from remaining 2 pathways. But only the first pathway is reversible pathway where glyoxylic acid is converted into glycine, whereas remaining 2 pathways are irreversible. So due to blockage of first pathway, which is reversible, the glyoxylic acid is not converted into glycine, and hence glyoxylic acid is accumulated in the cell, which undergoes further catabolism to form oxalate. So more glyoxylic acid is accumulated, so more oxalate is formed, and this oxalate is later on excreted into urine, and therefore this condition is called as primary hyperoxaluria. So enzyme deficient is alanine glyoxylate aminotransferase, which becomes inactive. Normally this enzyme is present in peroxisomes, but this enzyme in this disease, instead of peroxisome, is present in the cytoplasma, and hence it becomes inactive. So primary hyperoxaluria is a autosomal recessive disease, and here increase in the glyoxylic acid is seen. Now coming to secondary hyperoxaluria. Secondary hyperoxaluria is due to deficiency of coenzyme pyridoxal phosphate. This pyridoxal phosphate is again required for conversion of glyoxylic acid to glycine, the first reaction, which is transamination reaction, and pyridoxal phosphate is required. So as this glyoxylic acid is not converted into glycine, the concentration of glyoxylic acid is increased, leading to secondary hyperoxaluria, because later on that glyoxylic acid is converted into oxalate and it is excreted into the urine. Next disorder is glycinuria, inborn error of glycine metabolism. Here there is a defect in the reabsorption of glycine in the renal tubules. Normally all the amino acids which are filtered through the Bowman's capsule or through the glomerulus are reabsorbed back by the tubules, particularly the proximal columnated tubules. But in this case, remaining amino acids are reabsorbed, but glycine is not reabsorbed, because every amino acid reabsorption, they have particular transporter. Here there is defect in the transporters of glycine, and hence this glycine is not reabsorbed from the tubules. So more and more glycine is excreted into the urine, leading to glycinuria. Here plasma glycine level is normal. Now next is hyperglycinemia. It is also inborn error of glycine metabolism. There is defect in the catabolism of glycine, hence the glycine level in the blood, CSF and other fluid increases. Due to deposition of this glycine and its metabolites in the brain, it leads to mental retardation and seizures. Now we will come to the important products obtained from glycine. So this is the list of 8 important products obtained from glycine. We will study one by one. First is synthesis of heme. If you know heme synthesis pathway, then you can found that the first step of heme synthesis pathway is glycine combines with succinyl CoA to form delta amino levulonic acid, and the enzyme is delta amino levulonic acid synthase. Later on, this delta amino levulonic acid, after few steps, get converted into heme, and heme concentration when increase in the cell, it will inhibit this enzyme delta amino levulonic acid synthase to stop its further production by allosteric mechanism. Next important product obtained from glycine is creatine. Creatine is synthesized in the muscles. It is synthesized from 3 amino acids: glycine, arginine and S-adenosyl methionine. S-adenosyl methionine is nothing but it is a active methionine. If you see here, you can find that on the left side there is methionine which combines with ATP. This ATP is hydrolyzed, 3 ATPs are removed, and adenosine group of ATP is added to the sulfur atom of the methionine. Therefore, now methionine is called as S-adenosyl methionine. Due to attachment of this adenosyl group to the sulfur atom, the original bond between sulfur and methyl group becomes weak, and this methyl group can be easily detached from S-adenosyl methionine. In our body, there are so many acceptors are present which accept the methyl group. So this acceptor now accept the methyl group to form methyl acceptor. When the methyl group is removed from S-adenosyl methionine, the compound is called as S-adenosyl cysteine is formed. So in this way, S-adenosyl methionine and S-adenosyl cysteine undergoes transmethylation reaction. Coming to the creatine synthesis, first glycine combines with arginine to form guanidoacetate. The enzyme is amidotransferase. Now this guanidoacetate act as a methyl acceptor which accepts methyl from S-adenosyl methionine SAM and get converted into creatine. And after removal of methyl group, just now we have seen S-adenosyl methionine is converted into S-adenosyl homocysteine. Now enzyme required for this step is S-adenosyl methionine guanidoacetate methyl transferase. So both the substrate are written in the enzyme, and methyl transferase you have to add. Now this creatine later on combines with ATP, takes the phosphate bond from it and converted into creatine phosphate, and this creatine phosphate is stored in the muscle. Whenever body needs sudden energy during exercise or running, at that time this creatine reverse reaction will take place. Creatine phosphate donates its phosphate group to ADP to form ATP, and this ATP is later on utilized by the muscles. After reversal reaction or after donating this phosphate group, this creatine is converted into creatinine and excreted into the urine. So this is that I have already explained the role. This formation of creatine to creatine phosphate is a reversible reaction. During the exercise, this creatine phosphate is converted to creatine, and that creatine is later on converted to creatinine. Next fate of glycine is synthesis of purine nucleotides. Here I have shown the ring of purine nucleotides. During purine synthesis, complete glycine molecule is incorporated into the ring of the purine nitrogen base, where it forms C4, C5 and N7 of the purine nucleotide. Next is synthesis of glutathione. For synthesis of glutathione, you need 3 amino acids: glutamic acid, cysteine and glycine. So in this way glutathione is synthesized. Glutamic acid, also called as gamma glutamic acid, because gamma carbon carboxyl group is attached to this glutamic acid. So gamma glutamic acid combines with cysteine to form gamma glutamyl cysteine. For joining these 2 amino acids, energy is required, which is supplied by ATP, and the enzyme is glutamyl cysteine synthetase. Now this gamma glutamyl cysteine combines with phosphate to form gamma glutamyl cysteinyl glycine, which is also called as glutathione. Again, for joining these 2, energy is required, which is supplied by ATP. Next function of glycine is it is acting as a conjugating agent. Here I have given 3 examples. First is benzoic acid. What is benzoic acid? It is a preservative present in the food. It is partly soluble in water, therefore it is difficult to remove from the body. Glycine is an amino acid, and amino acids are hydrophilic substances. So when this glycine combines with benzoic acid, it forms benzoyl glycine or hippuric acid. Due to addition of glycine, that solubility nature of hippuric acid increases and it is easily removed from the body. Next is sodium cholate. It is a bile salt. Bile salts are synthesized by liver. They causes emulsification for fat, which is an important process for digestion of fat. Sodium cholate is also partly soluble in water, therefore it cannot be easily removed, or it cannot be easily excreted through the bile. When glycine combines with sodium cholate, it forms glycocholate, and due to addition of glycine, now glycocholate becomes more soluble. Next is sodium chenodeoxycholate, which is also a bile salt and partly soluble in water. Therefore, it combines with glycine to form glycochenodeoxycholate, which becomes more soluble bile salt, and therefore it is easily excreted through the bile. Next is synthesis of collagen, proteins and enzymes. Collagen is a protein present in the muscle. In collagen, every third amino acid is glycine. In various body proteins and enzymes, the glycine is present along with other amino acids. Next is glycine itself acts as a neurotransmitter in the central nervous system and brain. And the last is synthesis of serine. This is a reversible reaction that we have already studied during glycine synthesis, where serine is converted to glycine. Here, according to the need of glycine, according to need of the cell, glycine combines with N5N10 methylene tetrahydrofolate and water molecule to form a serine molecule. This N5N10 methylene group is added to the glycine and tetrahydrofolate becomes free. The enzyme is same, serine hydroxymethyl transferase. You can write down this reaction in this manner also. So this is about the glycine metabolism, where we have studied synthesis and catabolism of glycine, and we have seen various important products obtained from glycine, and we have studied various metabolic disorders associated with glycine. Here the hyperoxaluria is important, which may lead to renal stones due to excretion of more and more oxalate through the urine. Thank you very much.